Welcome, everyone. Thank you for joining. My name is Kate. I'm going to be your host for this webinar on multisensory perceptions and food-related decision-making. We're so happy to have you here, and we're especially happy to have Danni Peng-Li, our Customer Success Manager at iMotions, here today talking about his PhD research that actually led to a recent publication. So this is the first of our webinar series about bringing your biometric research study all the way from the study design process to collecting data all the way through to actually publishing in journals. So we're so excited that we actually have Danni here, who he is actually a former client of ours and is now coming onto the team and working with us as a customer success manager. So that's what today is about, is basically his journey and how he brought his publication all the way through the process, and then ending at iMotions is a little tidbit for the end. But let me just go quickly through the agenda. So we have Danni here. He's going to introduce himself a little bit in a second. Then I am going to intro iMotions to you all. If you haven't heard of who iMotions is, maybe you're not a customer, maybe you've heard about us, but you're not exactly sure what we do. I'm going to just give three quick minutes about what we do at this company, and then we'll hand it over to Danni, and we're going to have an informal sort of interaction and conversation. So there will be time for Q&A. So if you can see that your question box on your screen, you can enter a question for Danni. I will be queuing those up to ask him periodically. So feel free to put your question in. And yeah, this webinar will also be recorded. So everyone who has signed up, whether you're here right now, or for example, if you have a colleague who couldn't make it and they want to see the recording, everyone who is signed up with the registration link is going to get it tomorrow. So no worries about that. Also, if you have to jump off early, you'll get it in 24 hours. So with that, who's iMotions? We are serving global customers who are doing biometric research. We were founded in 2005, so we've been going for over 15 years, and we have offices globally. So in Copenhagen, which is where I am and where Danni is, our headquarters, and then we also have offices in Boston, Singapore, and Berlin. And we have a client base of over 1,300 clients, and that's both on the academic and the commercial side, which I'll explain in a second. But if you're curious about what biometric research is using biosensors, it's a way of investigating sort of in the moment physiological signals that our bodies send as they relate to emotion, attention, cognition, and also physiological arousal. So imagine that you're exposed to a situation, maybe something on a screen, you're watching a TV show, you're watching a movie, or you're even in an environment. So you're out walking along the street and you want to take in what your visual attention is, where your eyes are going, where they're looking. Biometric research and biosensors are a way to collect that data instead of just relying on what we call self-report, which is asking people how they experienced an environment or a situation or a stimuli after the fact, which tends to be a little bit exposed to bias. So we like to say that biosensor-based research can get you the sort of underlying insights about our bodies, what our bodies are kind of telling us about how they're reacting to a situation, in addition to pairing that with self-report through surveys, that kind of thing, which Danni will talk a lot about because it's related to his research as well. So we have a ton of different biosensors that we integrate, and what we do at iMotions is that we streamline that biosensor research using one platform. So we synchronize, for example, eye tracking, like I mentioned earlier, or facial expression analysis, which is automated detecting facial movements to detect facial expressions, along with GSR, which is galvanic skin response, for example, which Danni has used in his research, which collects data about sweat activity from the skin, and EEG as well. All of these different sensors, we combine them, we collect them, and you're able to design your study and present stimuli at the same time that you're collecting that data. Then we also have a whole platform of analysis and export options. So everything is synchronized into one platform, and it makes you basically able to do a one-stop shop type of research. And like I mentioned earlier, we power both academic and commercial labs. So you can see some of them here. And that's doing any kind of research from, for example, automotive research to commercial R&D, product development, but also a lot of academic research like psychology research, performance and training, sort of like helping diagnose disorders, healthcare, all of the different ways that you can imagine that your body might be able to tell you something about a signal that your body is sending you, we can basically do it. So whatever your use case is, we'd be super curious to hear what you're working on. You can always reach out to us if you have questions about whether iMotions is the right place for you. And on that note, I would love to start talking to Danni about how did you first get interested in human behavior research and iMotions? What's your origin story?
Yeah, first and foremost, thank you for the introduction. Super excited to be here. I think I've always been interested in psychology and human behavior, at least during my academic career. I think it started probably during my undergrad project where I was investigating the effect of fish oil intake, so omega-3 fatty acids on the cognitive functions in children. And that really sort of carried on during my master thesis, where I was able to also look into the both behavioral as well as neural correlates of food addiction. So that really grabbed my attention, and again, that just carried on throughout the whole PhD, during which I also had the opportunity to also explore how the environmental contextual factors could also affect our behavior. So not only internally, but also externally. So I think this is also this is also the point in time where I got introduced to iMotions, because my supervisor, shout out to Janice Wang, had actually at that time just purchased an iMotions license, and since I was her first PhD student, I had the freedom, the flexibility, the privilege sort of to play around the software and use it in my studies. And that made a lot of sense because my research was in this field of multisensory integration, perception. So I thought, what better way to investigate multisensory perception with multimodal research? Yeah, exactly. So that made a lot of sense. And yeah, the first time I actually got introduced to iMotions physically, sort of, you could say, is during these iMotions Academy I was attending back in 2019. And here I got, like, we were a big group who got a hands-on experience of like 5 days with the software and the associated hardware that was relevant for our own prospective research. So that was really nice to get really hands-on experience, and also to put a face on the people who has actually helped you along the way. And there was, of course, some social aspects with it, and you even get this very nice certificate proving that you have iMotions experience. So that was, I guess, my first real encounter.
Mm-hmm. Yeah, and were you sort of new to these types of sensors and this type of research? Had you encountered it before? You mentioned that Janice had just purchased an iMotions license, but had you been exposed to sort of biosensor research in the past, or was the Academy kind of your first hands-on experience?
I would say I had a little advantage, since I had played around with it in the beginning of my PhD. So I was playing mostly around with eye tracking. I think we had GSR in our laboratory, but the Academy, I was for the first time, physically playing around with other hardwares that I was not necessarily going to use in my studies at that time. So the Academy was, giving me, as I said before, a hands-on experience with also other hardware.
Yeah. Okay. Yes. Great. So now let's go into your study a little bit. I want to hear what it was about, and also for our audience to understand what your PhD research was that actually led to this publication, so that we understand how you came up with the idea, so that we can kind of get a preface into how you ended up getting it published. But yeah, so what was your study about? Where did the idea come from?
Yeah, so if I allow myself to zoom out a little bit, the overall objective of the whole PhD project was to explore how background sound could influence food-related decision making, and with a vast focus on these underlying psychophysiological mechanisms that you were mentioning before, which biosensors can capture. So that sort of set the overall framework for all the studies in my PhD, including the one I'm going to talk about today. So Katie, go to the next slide, please.
Yes. exactly.
Yeah, so this study here, which actually has just been published yesterday in the Journal of Frontiers in Neuroscience, and here
Congratulations.
explored the combined effects of noise level and cognitive regulation on the neurophysiology of food cravings. So basically, we wanted something that had robust experimental validity with these objective measures, which are not prone to subjective self-report and biases thereby, but at the same time also was relatable and applicable in the real world, sort of. And restaurant noise is something I would say everyone can relate to. So if you go to the next slide. So yeah, just an example here. I think most people have been in this kind of situation, having the choice between more palatable, delicious, mouth-watering cake and burgers as compared to the choice with more healthy, perhaps the more rational choice of, yeah, a salad, for example. But then imagine that you're sitting, in this restaurant, but there's just too much noise going on in the background. The music is just too loud. People are chattering, tableware is falling, and to make this example complete, babies are even crying. I think most people in this scenario would feel a bit distracted and perhaps even overwhelmed and stressed out from all these sensory inputs. And I think that would make it even harder for them to make the sort of rational choice for long-term health benefits for the food. So yeah, if you can go to the next slide, sorry. Yeah, so the theoretical foundation and motivation for all this is basically twofold. Maybe a lot of you here have seen illustrations similar to this, but basically there's been a large body of evidence studying this, what we call dual system model of decision making. It's of course very simplistic, but it's a very nice view, I think, still. Essentially, you can divide the way we make decisions into two systems. So one is the more fast, rapid, autonomic decisions, and on the other hand, we have the system two, which is requiring more cognitive resources, is perhaps more conscious. So we have the system one, which is fast and based on heuristics, and the system slower, deliberate system two. Yeah, and if you can please go to the next slide. And then in like another body of research, especially in the field of sensory marketing, there's been a lot of studies recently demonstrating that also these multisensory cues in the environment can affect our behavior, and that also includes auditory cues. So that could be background sound, music, and noise. There's been a few examples of studies demonstrating that just by turning the volume up could make people choose more unhealthy foods as compared to lowering the volume, which would make them choose more healthier options, which sort of set the framework for my study here. If you go to the next slide here. Yeah, so this is basically the whole conceptual framework. So we, in light of these two streams of research, yeah, this one, yeah, we tried to combine these. So we investigated both, the decision perspective as well as the noise level, and how those could affect food cravings, but also emotional arousal and cognitive load, as measured by galvanic skin response and EEG.
Yeah, sorry, I'm just checking to make sure everyone can see the screen, because we're hearing from some people that they can't share, or that they can't see the screen sharing. So I'm just checking, but it looks like it's fine. So sorry, keep going.
Okay, I do see the presenter view now, actually.
Yep. Yes. So yeah, if you're having trouble seeing the slides, I would recommend that if you're in the audience, try refreshing your browser and see if that works.
Yeah.
Cool. Thank you. Okay. Yep. Go ahead, Danny. I'm going to put back the slideshow.
Okay, perfect.
Yes.
Um,
Okay. Yes.
yeah, so I just finished on that note. So that was basically the study and
Yes.
motivation. Yeah.
Okay, great. So yeah, can you explain what you hoped to achieve kind of when, like, so you had thought about this restaurant noise and sort of this decision-making parameters, understanding like a real-world application for this type of sensory research. But what did you actually hope to achieve based on your study design? And can you talk a little bit about how you actually set up the study?
Yeah, yeah. So if you go to the next slide here. So of course we took something very, real life scenario and tried to put it into a laboratory, which is not always possible, but of course, in order to have these objective measures, it is necessary to do so. So what we did was to adapt this so-called regulation of craving task, which is a validated task that has been employed in various instances. And the picture here is just a trial example of the experiment we did. And as you can see, each participant went through two experimental conditions of the experiment. So one's with loud ambient restaurant noise and one's with soft noise. And then, as the picture here illustrates, for one trial, when they were exposed to either this is of course in Danish, so it may not make sense, but this is a now or later cue. So this now or later cue sort of describes or tells the participant how they should think about the preceding picture of the food. So for example, if they are shown the now cue, they should think about the cookies, for example, about the immediate sensation. So that could be the taste of it, the smell of it, and then after that they have to rate how much they crave that food on a visual analog scales. And then in the other case, if they're exposed to this later cue, and then are shown an image of the cookies, they should think about the later consequences associated with eating that food. So that could be the health benefits, or that they will gain weight from it. And then after that, they will again rate from a scale, from a visual analog scale, how much they then crave that food from that future later perspective. So the point here was kind of trying to make people either engaging the system 1 and the system 2 processes, while also having this manipulation of noise. So it's essentially a 2x2 design here. And if you
Right.
then and if you then next one, so yeah, this is just a picture of the setup in the laboratory. So as you can see, it's not in a restaurant here, but we're playing the noise through speakers in the background. And then you can see the participant here has a lot of sensors attached. You can see the EEG cap, which is measuring these brain waves and thereby cognitive load. We have the GSR electrodes, which could capture the skin conductance, and which is a measure of emotional arousal. And then on the right side here behind the wall, I could, as a researcher, then monitor live what is going on in the experiment. Yeah.
Right. Yes, and it's interesting here that, yeah, of course you weren't in a restaurant because you wanted to be able to work through to contain the conditions so that they were actually manipulatable, right, and controllable in terms of what the data was that you were actually picking up. And I think that's actually a great point to remind people of, is that especially if you are working with EEG and GSR as well, the skin conductance sensors, they are pretty prone to picking up noise artifacts. So having people in a sitting condition where they're really not moving is optimal for these two sensors. So if you're thinking about using EEG and or GSR, I highly recommend a lab setting. There is such thing as an ambient EEG way of collecting data, but it's much more prone to noise. So if you're curious about how to actually set up your sensors for this study, we have like amazing people like Danny working with us to advise you on the best study setup for you. Yes.
Yeah, I mean, it's always a trade-off when you are doing these studies. Sometimes, I mean, this is a trade-off between ecological validity, but also on the other hand, this, as you said, controllability in the laboratory. So you got to weigh that out, of course.
Yeah, exactly.
Yeah, I mean, this image here is just, this picture here is just to show that this is the measure of GSR, which basically measures here the phasic signal on top of the tonic signal, which is again this indication of emotional arousal. The next picture.
Yes.
Yeah, and again here, this is just to show you when I refer to cognitive load. In my case, I was measuring the brain activity in the frontal part of the brain only, and especially in the what we call the theta power spectrum. So in what is around 4 to 8 hertz, and this is also what is commonly referred to as the power spectrum density, so PSD. Of course, you can have different other frequencies of interest, but for the case of this talk here, I'm just gonna confine it to this one here.
Yes. Yeah, and I think we'll explain a little bit about how you can actually assess those signals using signal processing, specifically with iMotions R notebooks, but we'll touch on that in a second. But you can assess for power spectral density and frontal alpha asymmetry, but that's for another time. But here you have the decision perspective again.
Yeah. So I thought now maybe this is just to recap. This is because you asked. This is what we hope to achieve, and this is basically what we hope to achieve. I think this maybe makes more sense now. So yeah, we wanted to look at it from both a bottom-up perspective, so with the manipulation of noise, one could say, as well as this more top-down perspective. So using cognitive decision regulation, and to explore how those affect not only self-reported cravings, but also these underlying psychophysiological mechanisms driving this behavior.
Right. So I'm curious also about how were the reactions from your colleagues, because I feel like multimodal research is still kind of a buzzword in some, I guess it depends on the academic department that you're in. Maybe you've heard about this kind of research more than others, but did you have any sort of like curious reactions? People were like, man, that sounds interesting, from people in your research sphere, or were they kind of skeptical, or were they like, this is cool, I really support you? Like, what was the reaction when you said you were going to use a multimodal research design?
I think, first of all, on the personal level, I think my supervisor, of course, was very happy that I would play around with it, because she has just purchased the license, of course. But on a more, I guess, more global level, in the field of sensory and consumers science, like researchers has traditionally relied more on these subjective measures, questionnaires, visual analog scales, which of course provide a lot and a ton of valuable information. But this, of course, required that the participant are verbalizing the responses and has, this conscious introspection and post-stimulus presentation. And that's not always possible to get the exact responses right. And then many real researchers in this field have realized that these sort of objective implicit measures that biosensors can provide, that might be the way to go forward. That said, biosensors should, of course, not be a standalone product, but going multimodal with multiple sensors in conjunction with these conventional self-report measures, I think that can really provide some deeper and more like holistic insights into what is actually investigated. And I think this is also just reflected in terms of my colleagues, that they were really excited to having these biosensors, and now I know that the lab that I was working at, it has been expanding the past few years. So I think the future is bright in terms of biosensors, definitely.
Yeah, that's what we're seeing as well. We're seeing more and more academic departments reaching out to us because they're sort of like seeing how multiple sensors, like multiple data streams, can actually add to your research insights. A lot of people start, for example, with eye tracking, and then they build from there. And that's also totally valid, especially because, as you mentioned, we're not eliminating the idea of self-report, but we want to just add on to those insights. We get more in the moment data in addition to what people report to us. But we're seeing that more people are understanding the value of, for example, eye tracking and facial expression analysis, or in your case, EEG and GSR, right? So yeah, like I said, the future is bright. But so in terms of the next step, from data collection to results, I'm so curious about what you actually found. And then this is obviously going to inform the publication, right? Because you're publishing your results. And this can be a way for our audience to understand how you got to that path to publication. But let's talk about what you learned.
Yeah, so I think there's a lot of researchers listening here, so they all, of course, all know their whole process from study conceptualization to publication. And here again, after we have conceptualized the study, collected the data, and then again, so the third part here, signal processing, is of course a very important step when using biosensors. And here, since I have the iMotions license, we are able to use iMotions in-built algorithm to analyze the data both for EEG and GSR and other sensors if I had used those, which made it really convenient if you don't, as a researcher, have a lot of hardcore hands-on experience with the signal processing. So that was a great help. And then, of course, you need to go to the next level and export the data into whatever software statistical program you are using. From that, you can then interpret the results, which will be on the next slide.
Yes.
Yeah, yes. So this is just a snippet of the results we found. We just take it from an end. So in the top left corner, you can see these were the subjective cravings, so from the visual analog scales. And as expected, participants, when they thought about these immediate sensations about the food, so the taste of it, for example, then the cravings was actually elevated, and sound, the noise condition did not affect that. On the other hand, in the top right corner, for the emotional arousal measured by GSR, you could say the opposite pattern happens. So whereas for cravings, the top-down regulation sort of could have an effect, but here for GSR, there was more the bottom-up strategy. So the noise level manipulation that had an effect. So basically, participants, they were more aroused during the loud noise condition, which is not surprising, I would say. And in the bottom left corner, for the cognitive load, which was measured using the EEG and the theta power spectrum, you could see here there was an interaction effect. So there was no difference in the soft sound condition, but during loud noise and when thinking about these, future consequences engaging one's system too, the cognitive load was actually augmented, and that is actually, I think that's really interesting, that this is also what we expected, that require a lot of cognitive resources when there's a lot of sensory input going on and you have to engage your system too. This is also where the cognitive load is highest. And then something I haven't actually mentioned, this in the bottom right corner, this is a metric that iMotions can actually spit out. It's called frontal asymmetry. This is also a measure based on EEG. Basically, it's a measure of approach versus avoidance. So the higher the score, the more approach behavior the participant has, and vice versa with avoidance. But this is a very interesting result because this does not actually fit our hypothesis, because here one would expect that the approach behavior would be higher when they thought about the immediate rewards of the food, but we actually found the opposite. So there was a higher frontal alpha symmetry score when people were thinking about future consequences. I don't know why that happened. One could argue that it could be due to the fact that these motivational behaviors were just higher when they thought about perhaps the health benefits of not consuming that food. One could imagine. It could also just be a matter of a higher mental engagement, which frontal symmetry has also been correlated to. But I think it's just it's an interesting finding, especially just saying that these subjective measures do not always, they at some times they can be decoupled from the objective measures. Yeah.
Right.
Yeah.
But I think it's super interesting, these results, because you basically have these two signals that are kind of compiling data on top of each other to give you these different indications of how the noise level and the decision perspectives are kind of interacting with each other. And more or less what I'm gathering from these results is that, like you hypothesized, when you're kind of in an overloaded sort of sensory experience like a loud restaurant, it takes a lot more of your mental space to make a decision about your healthy eating habits, and you might be subjected to a little bit more of like the now cravings than you would be in a sort of more calm setting. So it does seem to have some sort of effect on how interested or how like engaged you could be in thinking about your healthy decision making choices in terms of food intake. Is that correct?
Yeah, exactly. I think my research in general has investigated something we can all relate to, something we actually already know. Like it's not rocket science. It's annoying with a lot of noise and so on. It's probably better to make a crucial decision in calmer environments. You wouldn't go study in the middle of a marching band or something. So, but instead, I'm more investigating. So we know it happens. I'm more investigating. Okay, why does this happen actually? What are the sort of underlying mechanisms behind these behaviors? And I think that's really interesting.
Yeah, super interesting. So we are actually going really quickly through the time. We are already halfway done. So I wanted to ask you a bit about the publication process itself, especially for the attendees here today who are curious about whether they can follow in your footsteps in some respect. So can you talk about how you actually got this research published specifically, and maybe if you have any advice for people who are watching?
Yeah, so I'm trying to depict the process here in this diagram that I stole from the internet. So the publication process for this particular study was rather conventional, with the whole peer review process. I think a lot of researchers, they have been through this process also, so nothing new under the sun really. In my case, for a PhD student, the situation might be different from someone more senior, because we have a strict timeline, of course, in which we have to publish. So for me, the choice of the journal was very important. Of course, for me, it has to be a rather acknowledged journal, has to be somehow impactful. But perhaps most importantly, the publication speed is crucial in this case. And I saw that Frontiers in Neuroscience had like relatively quick publication speed, and this article here was actually submitted to a special issue. I believe Frontiers in Neuroscience, or the Frontiers Journal, they call it as a research topic. But these research topics, special issues, the publication speed is usually faster. So I submitted this in, I think, end of November, beginning of December, and it was actually already accepted I think it was a few weeks ago, and now it just got published. So it was really, really fast as compared to other journals, which I know has years of publication speed.
Right. So that's actually a good tip for attendees that look for special issues or research topics within your academic discipline, because they might have, a specific spotlight on a specific type of research that you could kind of bypass the years-long process if you can see that there's a research topic coming up that's relevant. So that's really good to know. Let's go to the next slide, maybe. And if you don't have other input about actually publishing, I want to move on to the interactive poll. But do you have any, before we do that, do you have specific other kinds of advice for the publication process itself after you've done your study design and data collection? And while you're answering this, I'm just going to call out to people that you are welcome to start putting your questions in from the audience in your chat box so that we can ask Danny during the Q&A. But yeah, any advice or anything maybe you would have done differently, for example, had you had a second chance?
Let me think. I think perhaps with this study here, actually, if I allow myself to go back a little bit, because I have an array of studies where I have been using the biosensors. Actually, the first line of studies I was conducting using iMotions was eye tracking studies, and here I was, of course, new to the software. I was new to the field of human behavior research, so I didn't really know which direction to go. So I kept it simple and just stick to one biosensor, which was eye tracking in this case. Now that I know, what iMotions can actually do with the integration of multiple biosensors, I would probably have integrated more in these studies, because it would be fairly easy to just employ GSR or just facial expression, which is just through the webcam, which doesn't even require additional hardware. Because, of course, for my studies, I could investigate when and where and for how long people were looking at the piece of cake, for example. But I wouldn't be able to conclude that, the reason for looking at that cake. I mean, it might just be due to the fact that the cake was very weirdly looking as compared to the other food that they didn't look as much on. And here, facial expression could give me these insights, the emotional responses when looking at this piece of weird cake, for example. So I would probably have integrated more biosensors if I had knew better.
Right, and that's our whole sort of modus operandi here at iMotions, is that think about all the sensors that could be relevant for you, but don't necessarily overload yourself, especially if you're new to the technology, because you can sort of think you're collecting a lot of really great data if you just add all the sensors that you want. But then you could be overwhelmed in the data analysis process, for example, or you wouldn't necessarily know if you are collecting bad data, for example. So yeah, think about it critically. We're always available to help you with your research question and sort of study design in terms of which sensors to use. But it's great to know that had you done this study again, maybe you would have incorporated some facial expression analysis. Yes. Cool. So there's a lot of questions coming in, actually. I see a question from Ellen Aris about which journal it was, and it was in Frontiers in Neuroscience, correct? Just making sure.
Yeah, it was Frontiers in Neuroscience, and in the section called Decision Neuroscience.
Okay, perfect. Great. I'm going to launch our little poll, and while you're putting your questions into the chat, I can see a lot of coming in, so we'll start to sort them out and make sure that we get them asked to Danny and make sure we have time for all of them. But as I'm doing that, I'm just gonna ask how we can follow up with you, if at all, because we have a bunch of different educational materials and also intro calls that we can give you a live demo, for example, if you're interested in iMotions. So I'm just gonna launch that, and I wanna know if we can follow up with you. So I'll run that for a minute or so, and we're distributing it now. But while you answer that, we have a really cool question, or a comment actually, from Wenjing, who writes, I think your research is really interesting. Similar findings have been shown how noise can be harmful to surgical performance. Low, medium sound classic music can positively affect surgical performance, while loud music can negatively affect surgical performance. I think that's super interesting. And do you have any comments on sort of like the cross-industry insights that Wenjing is mentioning compared to your research?
Yeah, I think actually this whole multisensory field is not only confined to food research and in consumer science research. I think people have realized across different disciplines that the auditory cues, the auditory environment, the auditory architecture in that particular context is really important, and especially when thinking about how easy, how convenient it is to manipulate sound. Not a lot of equipment is needed as compared to other, let's say, visual cues, for example. It's just a matter of turning up and down or changing the music, for example. Yeah, so I think that's really, there's a bunch of anecdotal studies within also food science, of course. But I know that, for example, I've read a study about that noise also can impact like creative thinking. So not in the context of food necessarily, just in general. These cognitive processes. So I think that relates a lot to these, performance, which is of course also a cognitive construct. So that's really.
Yeah, super interesting. There's definitely a lot of applications for this type of research. Thank you, everyone who answered the poll. It looks like the majority of you have, so I'm going to close it now. And I just want to ask the rest of the questions to Danny, because there's tons coming in. So we have about 15 minutes left. Mahmoud is asking, how many subjects did you have in your study? And did you study using actual foods, or did you use pictures?
Yeah, so we had 37 participants, if I remember correctly. Yeah, 37. And the pictures we used was the food we used was images. There were images. So when I say multisensory, it was of course confined to two sensory modalities. So audiovisual we were looking at, and the images were extracted from this validated database where it has been standardized in terms of the angle and the lighting conditions and so on.
Interesting. Yeah, and again, it's really important to standardize all of that so we don't get subjective results. We also had a question about are the data systematic, meaning how many volunteers you had? You mentioned that because each person might have a unique result, right? So how are you creating a systematized data set?
I'm not completely sure if I understand correctly, but I'm gonna try to answer it anyways. So first of all, maybe one could imagine that the two noise conditions would have an effect. What we did was that, so it was a repeated measure or within subject design. So both, all participants went through both sound conditions and these conditions were counterbalanced and randomized across all participants. So that kind of rule out the effect of any what you call order effect, for example. And in terms of the exact measures, I don't know if I'm getting at the core of the question here, but maybe if there's been a lot of debate, a lot of questions in general about, for example, this response from GSR, where of course people are responding different. They have a different autonomic nervous system. But for example, in the case of GSR, as the image I showed previously, there are sort of two signals that are measured. There's the tonic signal, which is based on these individual differences, like how much you as a person sweat, temperature, and so on. And then there's the fasting signal on top, which also is depicted by these signal peaks. And it's basically only these peaks I'm detecting. And in my special case, I was for statistical analysis, I was looking actually at the peak probability. So I'm only comparing the sort of the phasic signals and not these individual differences that might be. I hope that answers the question.
Great. We have a question from Seba asking, can you share the link to the paper, which we definitely will. Everyone who has signed up to this webinar, whether you're here right now or if you had to drop off or if you're waiting for the recording tomorrow, we will include the link to the paper because it has been published yesterday, correct, Danny? So there is a publicly available link.
Correct,
okay.
correct.
Nice.
Yes.
Good. We will include that in the email that goes out tomorrow in about 24 hours with the recording to this, and you'll also get the link to the paper. Okay, so another question. You might have touched on this a little bit, but in terms of using food pictures for the study, they obviously weren't allowed to taste the food, right? Because you have a now or later condition where you're thinking about how the food may taste, and that is what you're looking into in terms of food cravings. But do you think that actually doing another study where you have them taste the food could be something you could work on, and do you think that would give a different result?
Short answer, yes and yes, definitely. A bit more elaborative answer is this would be an obvious next step, definitely. As I said before, this was confined to audiovisual, so it could also be audio gustatory and visual, of course, and also olfactory, so becoming really multisensory. The sort of challenge here would be the measure of for example, EEG, because as Kate said, these objective measures can be really sensitive towards, for example, movements, which would create a lot of artifacts in the signal that you have to account for, and which the algorithm are not necessarily capable of processing. But for example, like if the participant were to chew the food, that would just create a lot of movement artifacts, would definitely interfere with the signal.
Right.
But I know that some researchers has sort of tried to explore this anyways, but then it becomes not that natural. Like when you're tasting food, you're always chewing, you're using your muscle and so on. But some research I've seen are using, for example, droplets, where they basically are using a chin rest, and then they are instructed to put out their tongue, and then a drop of artificial, substance are, arriving on the tip of the tongue only, and then they are able to see what's happening in the brain. But this is, of course, not what is happening in real life. So again, this it's adding another layer of complication. But of course, you can always sort of design you out of stuff out of the way. So it is not impossible, of course, but there might be some challenges that one had to consider.
Yes. We actually do have some clients who are doing this type of research where your head is on a chin rest and they're doing sensory and fragrance testing. So it's like you said, it's such a weird experience, because if you want to isolate the reaction to right about the taste on your taste bud, but you're in this sort of like strapped into this EEG headset and like with your chin on a thing, yeah, what other interferences are you getting in that data set from people being like, what is happening, right? There's, biosensors can tell us a lot, but they're not the full picture. So, yeah.
Exactly.
Yeah. Okay, we have a question from Joanne who's asking, how long did it take to complete the study from design all the way through to data collection?
Oh, yeah. That's a really good question. I wish the process were sort of linear, which it was not. And it was the time of COVID, so that made a lot of complications, and I had to do a lot of like experimental medication to actually be able to carry out the study. I think roughly it took 1 year, plus minus a few months. That said, I spent more time beforehand on conceptualizing another study that did not end up getting carried out at all, an fMRI study, actually, but due to COVID reasons, I couldn't do that. So I was actually trying to translate this already designed fMRI study into an EEG study, which you of course cannot do one-to-one, but there's a lot of the same considerations you have to take. So of course I was a step ahead in the process. 1 year ago, I already had the main idea. The challenge now was just to make it fit for an EEG system instead. But yeah, 1 year give or take. It was yeah.
Yeah, it's a long process, but the results that you've uncovered are, like, I hope, worth the time, because it seems like, especially because
I didn't spend one dedicated year for only this study, because as a PhD student, you need to be able to multitask, and you have different projects overlapping, and sometimes you are setting aside more time for other stuff, of course, but overall just 1 year.
Yes, right. Yeah. Great. We are quickly running out of time, but I have a question from Seba again, who says, since I work in the spatial hearing research, just wondering your thoughts on the following. Which differences you might see in your results if you create a realistic 360 acoustic scenario rather than a not natural 2-speaker setup, so kind of like recreating that restaurant vibe. Do you think that would maybe affect the results? And also, what particular noise did you use, and how was that standardized in the study?
Yeah, that's 2 really good questions. For the first question, I haven't given that too much thought, to be honest. I don't know if it would necessarily make a difference in the responses, but it would definitely make it more realistic, which would, of course, strengthen the robustness of the design and make it more like ecologically valid, one could say. In terms of the noise, we used this free sound .org is a database with a bunch of different soundtracks and soundscapes, and then we standardized it using an audio software. I'm sure that the person asking this has way more expertise than this. But we basically adjusted it through the it's called LUFS levels, I believe, to standardize the 2 different noise levels, so they on the same sort of scale. I have in the publication, there's a more detailed description on this that is possible to read if it's interested. Yeah.
Okay, awesome. Yeah, and also in iMotions, when you are presenting your stimuli, you can do, and I think you mentioned this when you were talking about study design, you can do your block like study design. Can you explain that a little bit, so you can make sure that you are sending the right blocks of content at the right time, and also standardizing what the stimuli is and how it's exposed?
Oh yeah, of course, yeah. So this is of course not only related to the audio, but just the general design stuff. Yeah, yeah. Yeah. Since this was just a rather simple 2x2 design, I could, for example, I could inside iMotions software, I could have let's say a block called soft noise and another block called loud noise. And then inside the iMotion software, I can just tell it to randomize these across participants. And then I can, of course, see when, whenever a new participant is coming in, what stimuli should he or she have. So it's just, you know, a complete randomization across participants, which
Right.
is inside the software.
Mm-hmm. Great.
Yeah.
Cool. So one last question before we close, but before I ask you this, I just wanted to mention that if we didn't get a chance to answer your question in this webinar because there were so many, thank you all for your amazing input. You can always email us at marketing@imotions.com, and we'll be sure to pass the question on to Danny so he can answer it individually for you. And yeah, I also want to mention that we do have a lot of publication webinars coming up. So stay tuned on our newsletter channels and in our yeah, all of our social media channels. We'll be announcing those as they come up, but the next one should be in a couple months. So just stay tuned for that. But if you yourself are interested in, if you have used iMotions for publication and you would like to amplify your research in this scenario, in this webinar setting, please reach out to us also at iMotions, marketing@imotions.com, and we'll be happy to work with you to prepare a presentation just as Danny has done. So yes, one last question before we end, which is, let me just read through this. Did you correct for ocular artifact in the EEG data?
No, we didn't actually. We used a very standard procedure using the iMotions algorithm. We referenced through the, I think it was the TP9. So not the not the ocular, but here behind the mastoid. So it's very simple setup. We did apply. It was a 32 channel sensor, so the sort of scalp resolution was rather high. But as I also mentioned in the talk here, that we ended up actually only focusing on the frontal part of the brain. I could of course have also explored other areas of interest, but for the sake of simplicity, we kept it to the frontal part, because for each region we also have there a bunch of different frequency bands, and you can always add more analysis, but again, it's a trade-off. So, but yeah, to answer the question, we didn't have that.
Right. And it is always a trade-off based on, the resources that you have at your disposal and the time frame and your analysis capabilities as well. But yeah, we always, again, at iMotions are available to help with EEG studies. We have experts like Danny on our staff to help you out if you are an iMotions customer. And if you aren't an iMotions customer, there's plenty of ways for you to get in contact with us. So if you go to imotions.com/contact us, we can set the gears in motion to see if biosensor research is right for you. Yeah, and on that note, I want to say thank you so much to Danni Peng-Li, PhD, for an awesome presentation about your research and your path to publication. And if anyone would like to follow in Danny's footsteps and do a research webinar with us, we would be more than happy to hear from you. So thank you so much. Have a great rest of your day.
Bye.